it's more informatics oriented. The title is Automated Metadata Ingestion and Harmonization in Strand's single-cell RNA-seq portal and beyond. Speaker is Badri Paduka Sahasram. He's the VP of Data Science. He heads our data science team at Strand Life Sciences. Badri brings over 2 decades of experience at the intersection of bioinformatics and data science. Worked at Roche and Illumina before this. He earned his PhD from USC in computational biology, postdoc at UC Davis. At Strand, he leads the data science group that designs algorithms, statistical models, and scalable data analysis pipelines for omics data. And the emphasis is on harnessing foundational models. Over to you, Badri.
Thanks, RK. Welcome to the Bay Area BFX Forum, and thanks for having me as a speaker. So today I'm going to talk about an LLM-based automated metadata ingestion and harmonization pipeline that we developed in the context of Strand's scRNA-seq portal, and we show that this pipeline can help ease the burden for our curators and speed up the process of data curation, which can subsequently support downstream research questions. So there's a vast amount of omics data available. Of course, the focus of the Strand scRNA portal is single-cell transcriptomic data, and the primary source is the gene expression omnibus. So a lot of the work that was done in the pipeline is developed in the context of this particular repository, but we show that the same methods or tools can also have broader utility and can be adapted to be a more general purpose tool. So there's a range of omics data and that can help answer a variety of research questions. And one of the issues, general issues with public data sets is with metadata heterogeneity. So the names of the fields of interest are not always present in a consistent fashion, and there is quite a bit of variation in expressing the same entity. For example, something like a CD4 positive alpha beta T cell might be referred to as a helper T cell, or something like chemical colitis can be referred to as toxic colitis. Type 2 diabetes might be referred to as hyperglycemia, and so on. So this type of varying terminologies can make it more difficult. So even before the technical challenges of integrating multiple data sets, issues like batch correction, just this inconsistent metadata is a barrier to integrating across datasets across the studies, which therefore hinders subsequent downstream applications. So this entails or motivates the need for metadata harmonization methods so that datasets can be integrated to maximize our power to answer research questions. So a lot of work for metadata curation prior to the development of our automated pipeline was done in a manual fashion. So it was done with a set of curators who would search the geo for the relevant studies, download the data, and then map it to certain standardized ontologies or dictionaries. So this process can be quite time consuming. So this is our portal, Strand's scRNA portal, which has a large collection of single cell data from numerous samples. So the focus is on inflammatory bowel diseases as well as certain neurological diseases. So there's a wide range of metadata fields, and there is multiple levels of curation at study and sample level, and a range of different filters. But the problem with manual curation is that it's quite laborious and time consuming for the curators, requires substantial investment of time and resources, and scaling this up to a larger number of studies or a larger number of metadata fields becomes challenging. This motivated us to develop this RAG LLM based AI pipeline that can help automate this process and ease the burden for our curators. The advantage of having a manually curated starting data set in our portal is that it serves as a ground truth against which we could benchmark our automated pipeline as well as perform further refinements and optimizations. So, how can we scale up or automate the metadata curation? We propose the use of AI large language models for this purpose. So the fields that are to be harmonized, they are of two different types. Like 19 fields have to be mapped to certain ontologies, and there is another set of fields that have to be mapped to certain internal standards. So here I'll illustrate the benchmarking and assessment for 8 of the ontology related fields, and we showcase the performance of our pipeline and the accuracy metrics for the pipeline. In terms of like approaches to automate the process of metadata harmonization, there are several different approaches that have been proposed in literature. These include use of pure large language model based methods, as well as use of mathematical approaches leveraging vector embeddings and distance between vectors, and as well as approach that use some combination of these two approaches. The approach we are taking, referred to as the RAG approach, is a combined approach that leverages both semantic or numerical representation of words or phrases, as well as the large language models. The different methods illustrated here. The first method is the most basic method, zero shot recall, where we are just providing the phrase of the natural language term of interest to the large language model and relying on its pre-trained knowledge to get us the answer. Then the next approach is prompt recall, where in addition to providing the phrase of interest, we are also providing it the dictionary of the normalized terms, and the large language model can pick the appropriate term from the dictionary. Of course, as it is understandable, as dictionaries become large, this can be time and cost intensive. Then we have a pure or more numerical type of approach where we rely on vector representation of words or phrases. So this is referred to as semantic search. So we convert phrases into vectors and also the dictionary of possible normalized entries into also a vector, and then pick the term which is closest in terms of some distance measure between the two. The cosine similarity is a commonly used distance metric. And lastly, we have the RAG approach, which is what we have undertaken for our pipeline, which combines the strengths of both prompt recall and semantic search. It uses semantic search to first narrow down the candidates from a dictionary. So instead of the entire dictionary, we are only taking the terms likely to be candidate answers for the term of interest based on that similarity. And then the LLM model is prompted to pick the most appropriate answers among the top k entries as determined from the distance metrics. So that's the general idea of the RAG approach. Um, so um, there are different steps or questions to be answered. So first is how do we convert the phrases to a vector representation? So there are different choices of embedding model. So one question is how to choose or pick the embedding model, and then there is also the question of what is the appropriate choice of k? How many top k matches should we take among the terms from the dictionary, so as to get the good good performance. These are two questions, and also the question of how to prompt an LLM to get us the answer we desire. So these are things that need to be addressed. So to address the question about the choice of semantic embedding, we compared a range of different semantic models, both natural language embedding as well as embeddings that are developed more specific to clinical and biomedical text and sentences, and we compared the performance on a certain test data set. So these embeddings vary in their dimensionality, and they can range from, for example, 384 to 4096, which also somewhat affects the runtime and storage requirements. But our comparison indicated that among these embeddings, a particular clinical and biomedical concept specific embedding, this BioLart performed the best, and we picked that as our embedding of choice. We also did further optimization with variants of BioLart, and while doing this optimization, we also varied the choice of the number of top k terms, the choice of k, which is the number of top hits we want to consider or provide as input to RAG. So both these choices were jointly optimized on empirical data, and we picked the optimal combination based on the performance. So overall, when implementing a RAG pipeline for our geo portal, there are some more complications or refinements that are needed. So one aspect is this issue of entity extraction. So the entity that we want to harmonize or normalize, it's not always directly available in a particular place in geo among the summaries. So sometimes it is within the summary paragraphs or description paragraphs. So one additional step that needs to be accomplished is first extract the entity of interest from the paragraphs, and then subsequently do the harmonization to an ontology. So we accomplish this also through the use of LLMs. So we have this upstream LLM that does the extraction of the entities, and here also how to prompt the LLM to extract the entity. That process also needs to be optimized. So in addition to optimizing the embedding model as well as the RAG parameters, we also undertake extensive prompt engineering to optimize or get the best possible results. We did all this, and then we evaluated performance on a collection of study IDs from the geo portal, and we found that after optimization, this type of a RAG plus large language model framework can be quite effective for automated harmonization, and we found that the accuracy is quite high, over 95% for most of the fields, and this suggests that it may be feasible to automate this manual process and laborious process of metadata curation. So overall, this approach provided promising results. Of course, one issue is that the LLM or the automated pipeline has some probability of making mistakes, so it's not fully accurate. So there's some chance of errors. So then the next step is for a subsequent manual check may need to be applied to maybe correct those errors. So the curators might be interested in doing a manual follow-up on the results from an automated pipeline. So to help prioritize this process, we also provide measures of quality for each of the normalized entry. So in addition to providing the automated harmonized term for the entities of interest, we also provide quality flag for each entry that gets harmonized. So this is accomplished through the use of independent evaluator LLMs that look at the input and the answers provided by the pipeline's LLM, and then provides a quality score in the range of 1 to 5, and eventually a quality flag indicating whether something is of low quality or good quality. So the idea is that the curators can focus their manual checks on the low quality flagged entries, which is a smaller fraction of the total number of entries, and do any corrections if necessary, so that the overall burden is significantly reduced compared to looking at everything manually. So here we demonstrate the just a validation of the quality control metrics. So we generated simulated examples with correct answers, as well as we simulated examples with plausible but incorrect answers, and then applied our quality score metrics on it, and looked at the difference between the quality scores. So the top row false is where the entries or the answers are correct, and the bottom row is where we have simulated different types of errors, and we can see that the quality scores is able to distinguish between the two categories. So the top row quality scores tend to be high, more towards the 5 and little on the 4 values, whereas the lower rows with simulated errors, there is a range of lower quality scores. So this suggests that the quality score metric is behaving as intended, and we have shown some precision and recall estimates based on our simulated data to illustrate the utility of the quality metrics. And lastly, we also did some comparisons with some prior implementation of RAG that relied on more natural language embeddings, and we found that our pipeline matches in performance. So generally, it appears that the biomedical specific embedding is working slightly better in terms of accuracy. Then here we illustrate the improvements in the turnaround time. So we have around 4 to 5x reduction in turnaround time compared to manual curation, and the cost is around 40 cents per data set. So all this was done in the context of the Geo portal and the Strand's scRNA portal, which was using Geo as the primary source. But so this tool that we developed is integrated with other tools that download the data from Geo and subsequently do this automated metadata harmonization. But the users may also be interested in more standalone normalization tools that is not so much linked to a particular repository. So we are undertaking additional work to develop a more general purpose standalone tool that can implement the same algorithms as in this pipeline, but for data text, more general text from the wild other than geo. And the users may also desire more flexibility in terms of the choice of entities. They may not necessarily want the entities from the geo. So we are working on this standalone tool to support entity extraction and normalization from any text in the wild, and we hope that eventually we will be able to support a wide range of entities for this standalone tool. To illustrate performance, we also tested paragraphs from another repository, the CellxGene repository, for which we had some summary paragraphs as well as ground truths available for certain entities. And so this is preliminary results from those comparisons, and we can see that the pipeline can produce reasonable results for text from other sources, not necessarily from geo. So to conclude, we have a high quality curated portal with a focus on IBD and certain neurological diseases. It has multiple levels of curation and a range of different metadata fields and filters. We have implemented an automated pipeline based on RAG LLM, leveraging AI foundational models, different variants of GPT, and this decreased turnaround time by around 5x compared to manual curation, and while incurring a cost of about 40 cents per dataset. So while LLM normalization has high accuracy, so there is some chance of error for each of the entities. So to aid subsequent manual checks on the pipeline, automated pipeline outputs, we also flag the entries with quality scores. So the idea is that the curators can focus on a small fraction of entries that are getting a low quality score, and fixing those should be able to capture most of the errors, as we demonstrate from the evaluation of the quality score metrics. And last but not least, we found that at least in Geo, not every field requires an LLM. So suitable combination of both traditional scripting and LLM can help scale up the problem of metadata curation for metadata ingestion and harmonization for public data. So I'd like to acknowledge the team, both the algo, data curation, as well as the software teams that contributed to this work. Thanks for listening.
Great talk. So I used to use Geo for my thesis, and I also had to do this curation work. So one thing I was wondering is, do you take into account the type of organism? So I know you mentioned that you could differentiate between like a mouse and human, right? But what about between like different mouse lines? Because that could be very important, if especially if you're looking like transgenic versus I don't know what other, right?
Yes, yes, definitely. I think the cell lines and the strain are all fields in the list of fields that I was showing. So definitely, those are details we want to capture and harmonize in as a metadata field in our studies. I think this process is also done. So there is like a study level information. There is also like individual sample level information within a study. So we offer two different levels of curation. So individual samples may vary in their strain details or the particular cell line details. So all that is captured by this pipeline. So it will harmonize the strain and cell line type of information as well. Yeah.
One more here.
Yes.
Great talk. I wanted to ask about the accuracy. So I think you had a slide that mentioned that the accuracy was around like 89%, right? Or 80%. I don't remember exactly what the number was, but I wanted to know, like, especially in like science, the level of accuracy is like really important. So how much does having like a 90% accuracy affect like the actual retrieval of accurate like data that scientists can actually use?
Yes, definitely. I think it's definitely a valid question. So I think as I was showcasing the performance, so I think our accuracy numbers for the pipeline are, I think, around at least for the Geo portal, those are around 95% or above. So if you see this table, so you can see that most of the fields, so we have been optimizing various choices of parameters and settings to maximize the accuracy of our data. And then the idea would be that we also have a mechanism to flag possible low quality answers. So those can be subsequently checked by manual curators to further improve the accuracy. So it will be it was some amount of manual effort, but it is only a small fraction as compared to manually curating the entire data set. So that's the type of workflow we are hoping to follow here. So this in addition to the original results, we will do a manual check of the low quality entries. But of course, ultimately, we want to do it even that in an automated fashion. That means have iterations of LLMs or agents acting upon the flagged entries. So that is something we are considering in future versions of the tool, to look at the low quality flag, then iterate or run other alternate LLMs or alternate prompts for those entries until we get high quality scores. So this type of iterative steps of the RAG LLM can further improve the accuracy from this single step accuracy that I'm showcasing here.
Yeah, we're taking two approaches. One is, of course, Badri's team is working hard on the agentic approach and can push the barrier on the accuracy. And I don't think this is meant to obviate the manual process. It's meant to reduce the amount of time you spend on it. So the target is always to reduce the amount of time, but not necessarily fully remove it. It's probably at this point not feasible to do that. One more question here.
Yes.
Hi, great talk. So I saw that some of the, in your next slide, probably some of the simulated errors also had a pretty high quality score. So you said that you'll flag low quality ones, but do you have any plans to get around these as well? Because manually people will probably overlook the high quality ones and focus on the low quality ones, but yeah.
Yes. I think we'll continue to refine the prompts we have designed for the quality scores. So I think definitely, I think this is our first iteration of a quality score, which is relying on evaluated LLMs. We also hope to further refine the prompts, of course, but then also add additional numerical metrics that could be indicative of LLM hallucinations, like things like entropy measures that have been published recently in literature, as well as other numerical metrics to supplement the LLM provided quality scores. Yes, definitely we would want to further improve and refine these metrics.
And there are two more. Do we have time, Shane? A couple more questions in the audience. So. Francisco.
Thank you. One of the problems real data has is that some of the fields are missing. And traditionally what we have done is use statistical imputation methods to cover those missingness, right? We were talking about accuracy, but sometimes we have missingness, significant missingness. Have you started thinking on using LMS as a way to impute missing values and enhance data sets as well?
I think that may I think that may you need the use of also in addition to LLMs, maybe machine learning approaches as well, some type of deep learning architecture. So we haven't thought about this particular type of issue. So generally, what we find is that there is a lot of missingness in the geo portal, and sometimes the relevant field or entity actually resides within the publication or the supplementary section. So we are working on improving our tool to be able to ingest entire publications and then look for the missing fields. And that's something we are planning in the next iteration of this pipeline. But your problem itself, yes, I think it's an interesting problem. Data imputation, whether it can be we can use leverage. Yeah, definitely it's an interesting direction. I would like to explore that further, but so far I haven't thought much about that. My first thought is maybe some type of deep learning or transformer models with the data as input, with like data having full data and some data having missing data can be used to train the data with full set of fields can be used as the training model to impute for the data with missingness. But again, I haven't really thought about it. It's an interesting idea. I will consider it and think more about it.
Beyond some symbolic semantic links, have you looked at context and symbolic type links or use for the RAG agent? For example, you just mentioned looking at the rest of the article for the geo context. and using the context within there to help you extract the right terms or to
Yes,
match
yes.
the right terms.
Yes. So I think there I was thinking more the information is, so I think what I was referring to is that the information pertaining to a field is not directly available on the geo. So for example, in single cell type is an key or fundamental entity. Usually it is available within the publication, and many times it's missing on the geo summary. So I think what I was talking about is just ingesting that publication, and so getting the missing information from within the publication. But yeah, I think we are using reasoning models in some of our entities where the context is important. So for example, when we are trying to figure out maybe some status like a disease status at a sample level, definitely we are using the context. So like what type of information, if it mentions a control, or if it mentions healthy, or if it mentions a particular disease, so all that is taken into account, which organism was studied in that particular sample. So all the context surrounding the study, so we combine a series of paragraphs from many different sections of the geo, and that is provided as input to a reasoning model to leverage and make better decisions in terms of identifying certain entities. So definitely we are making use of that currently, but in the in publications as well, we will, if there is information that can help us infer certain fields, definitely we would make use of it in the future.
Thanks all for the discussion, and thank you, Badri, for your time.